you might have wondered how each and every one of you have one or the other characteristics that you share with either of your parents. It might be your facial structure, your height, your weight, etc. But have you ever wondered about the molecular basis of this heredity? Well, that is what I'm here to explain today. Good morning to everybody present here, respected judges and my fellow presenters. Today I'll be presenting the topic of nucleic acids and a deep dive into their structures as well as their functions. So what are nucleic acids? The nucleus of a living cell transmits heredity through something we all know, chromosomes. And chromosomes have mainly two components. One of them is proteins and the other one is nucleic acids, the topic of my presentation. Nucleic acids are long-chain polymers of nucleotides or also known as polynucleotides, and they are of two very popular common kinds, DNA as well as RNA. Now, hydrolysis, as in when we split the nucleic acids, we get three parts to it. One of them is the pentose sugar, second phosphoric acid, and the last one is the nitrogen base, which we'll be deep diving into the next slides. But firstly, the pentose sugar has two main differences in DNA as well as in RNA. We can see over here how the hydrogen group is replaced by the OH group in the sugar of the RNA, which is the beta-D ribose sugar, and over here in the DNA, which is beta-D2 deoxyribose sugar, has only a hydrogen group instead. Well, as I said before, nitrogen bases are also a very integral part of nucleic acids. And there are 5 main nitrogen bases, and uracil and thymine over here are not present in both DNA and RNA. Uracil is present in RNA, whereas thymine is present in the DNA molecule. Firstly, we have adenine and guanine, both of which are purine nitrogen bases, and cytosine, uracil and thymine, all of which are pyrimidine nitrogen bases. So the basic difference over here between purine and pyrimidine nitrogen bases is that we have two rings in purines. One is the purine and the other one is one is a pyrimidine and the other one is not a pyrimidine ring. So that is the major difference between both the pyrimidine and the purine rings. So here is a small animation to show how polynucleotides get made. We see over here that the phosphate group attaches itself with another pentose sugar group of another nucleotide, and we can see over here that this 3' and the 5' of both the pentose sugars are involved in this reaction. This bond is known as a phosphodiester linkage, and this occurs due to condensation between the two H2O molecules that are formed in pentose as well as the phosphate group. So we can see over here that the 3rd carbon as well as the 5th carbon of the other pentose sugar create this bond, which is known as the phosphodiester bond. So the sequence of the nucleic acids in a particular DNA or an RNA form its primary structure, which is very crucial because it tells us how to differentiate between different nucleic acids. But the secondary structure was also observed further by two particular scientists, Watson and Crick, which introduced a double-stranded helix structure for a DNA, as we can see over here in the model as well. Two nucleic acid chains are bonded by hydrogen bonds between the two bases. In my model over here, we have the nitrogen bases over here as adenine and thymine, guanine and cytosine, which form bonds with each other to make sure that two nucleic acid pairs are bonded together. So adenine and thymine have a specific of double hydrogen bonding between them, whereas the cytosine and guanine have a triple hydrogen bond between them. And as I said before, thymine is replaced by uracil in the molecule of an RNA. Okay, so for a DNA, we know that it's a long molecule made up of many monomers known as nucleotides, deoxyribonucleic acid. Let's break down this term. So the deoxyribose component stands for the sugar that we had discussed in the second slide, whereas the nucleic acid is the macromolecule that our topic is based on today. So the phosphate, the deoxyribose sugar, and the base together form the nucleotide, and many nucleotides together form either the DNA or the RNA. So we see there is a twisted ladder shape known as the double helix when it comes to a DNA. Let me explain this model further. We have one nucleotide with a phosphate group with another bonded with a pentose sugar and a nitrogen base. This nitrogen base forms a hydrogen bond with another nitrogen base, whereas the phosphate and the sugar form a phosphodiester bond between them. So as you can see, phosphodiester bond is stronger and it makes the backbone of the DNA through the bonds between the phosphate and the pentose sugar, whereas the 4 nitrogen bases are held together by weak hydrogen bonds that are very weak compared to the phosphodiester bond. Here we can see the difference between the structure of a DNA as well as an RNA. The main difference between both of them is obviously the thymine and the uracil component that is not present in either of them. And deoxyribose sugar and ribose sugar, which is also different in RNA as well as DNA. So I have discussed regarding the secondary structure of a DNA, which is introduced by Watson and Crick, which is the double helix structure. But what about the secondary structure of an RNA? Well, RNA folds by itself, on itself, to create a secondary structure different from the primary structure, which is the sequence. And over here we can see that RNA has multiple secondary structures, 3 of which are very crucial in the process of protein synthesis in human beings and other biological processes as well. As I stated before, the 3 main RNA components that we have are mRNA, rRNA and tRNA. So decoding mRNA, which is the messenger RNA, is a single-stranded RNA that corresponds to the genetic sequence of a gene. So this will be more in depth for the biology students, but in chemistry all we need to know is that this helps in understanding the sequence of a gene so that it can go forward to code it. And it is read by the ribosome in protein synthesis. And we have the rRNA, which is a non-coding strand that is only a primary component of ribosomes, and it can be seen in this particular picture where there is a large subunit and a small subunit and an mRNA helping it to read whatever is there in the DNA. Lastly, we have the tRNA, which is the transfer RNA and which is one of the most important parts of RNA during protein synthesis. It attaches itself between the messenger RNA as well as the molecule, and it helps in the growth of the amino acids that will be attached according to the codons. Finally, though we have discussed so much about DNA and RNA, one must be wondering what is the actual biological function apart from protein synthesis. We know that DNA constitutes of the chemical basis of our heredity. It serves as a reserve for a lot of genetic information over the many years that we live, and it also helps for cell duplication during cell division or transfer of DNA strands to the daughter cells as well. And obviously there's protein synthesis by the RNA molecule and the messaging duty that's considered by the DNA molecule. Thank you for listening to my presentation. If there are any doubts, I'll be glad to answer them.
Can you please explain what are the black and white bonds in the structure?
Yeah, sure. So, first of all, dividing the structure into half, this one particular component is one nucleic acid. Okay, the black part is the phosphate. The phosphate group that is attached in the nucleic acid, and the white part is the pentose sugar. So we know that the phosphate and the pentose sugar is attached together, and this pentose sugar is further attached to a nitrogen base, which can be either of the 4 types in DNA or replaced by uracil in the RNA. And this one is called a nucleotide. When it attaches itself, it's a polynucleotide, and a long chain of polynucleotide is called a DNA. Hope that's clear. So over here we know that all pyrimidines have only one single ring, which is known as the pyrimidine ring. But over here we have 2 purines, which is adenine as well as guanine, which have 2 rings. They also have the pyrimidine base ring, but it is accompanied by an imidazole ring, which is an attachment present in adenine as well as guanine. So that is the primary structural difference between adenine and guanine compared to cytosine, uracil and thymine.
Can you explain why DNA is more stable than RNA?
Okay, so basically DNA is more stable than RNA because one primary component, which is uracil, causes an instability in the RNA molecule compared to the DNA molecule. Apart from that, it is also the existence of this particular OH group in the sugar component of the RNA, which causes it to be very unstable during reactions and other procedures. Hope that's clear.
So you mentioned folding of RNA. So can you explain
Yeah, sure.
a bit more?
Yeah. So basically, as I stated before, all nucleic acids have a primary structure which constitutes of their basic sequence. So if that sequence is changed, the nucleic acid completely changed. But from the primary structure, we have a secondary structure as well. But for DNA, it is easy to determine the secondary structure since there are two strands contributing to it. But in the case of RNA, the same molecule of RNA folds over itself. As you can see, there are many other examples of RNA folding over itself to form new structures to aid in various processes. So each of these RNAs over here, as you can see, the single-stranded RNA, the 3-nucleotide bulge, the single nucleotide bulge, all of these are various structures formed by a single strand of RNA folding back on itself and creating new bonds for various purposes. Yes.
Could you go what is phosphodiester bond?
Yeah, sure. Okay. So basically, as we know that there is a phosphate group in a nucleotide that is present. So this phosphate group, to attach with another nucleotide on top of it to form a double helix structure, creates a bond with the pentose sugar. Okay. So this pentose sugar and bond between the phosphate is called a phosphodiester bond, and it is formed by the removal of H2O molecule while this bond is formed. So it's also known as a condensation reaction when there is phosphodiester bond. This animation will further help you. So if there is a sugar molecule and a phosphate molecule, these two molecules want to attach themselves with the sugar molecule and the phosphate molecule of the DNA, the nucleic acid above them. So when this bonding happens, that's when a phosphodiester bond comes into place. So this is the phosphodiester linkage.